Okay. So I'm starting it. Hi, this is Dr. Amna. I'm starting up with the paper 970012. It's February, March 19, 2024. So let's see how we go about. This is our first question. Okay, so let's read it. You can read it by yourself too. I'm also reading it for you. A student used a light microscope to observe a blood smear on microscope slide. An eyepiece graticule was used to measure the diameter of white blood cell on the slide. So whenever you are doing the MCQs, I would suggest to you that it's better that you underline the keywords, okay? So over here, the first keyword I'm finding it over here is the eyepiece graticule, okay? And we want to measure the white blood cell diameter, okay? The student recorded the whole white blood cell was 5 eyepiece graticule unit in diameter. Okay. I have been given with 4 choices. The first, so they're asking which additional information does the student need to determine the diameter of the white blood cell in micrometer. So I need to determine the diameter of the white blood cell, and this is the set of information I have. So what next I need to determine the diameter? I have been given with 4 choices. Calibration of the eyepiece graticule using a stage micrometer only. Calibration of the eyepiece graticule using a stage micrometer and the magnification of the eyepiece lens. C, the magnification of the eyepiece lens only. D, the magnification of the eyepiece lens and the magnification of the objective lens, okay? So what I see over here in B, C, and D, I find the word magnification, okay? And when I am finding the actual diameter of the white blood cell, we do not need to know the magnification. So over here, these 3 will definitely be wrong. A is the correct one. I want to explain a little bit more about the stage micrometer and eyepiece graticule and how we do it. Just give me a moment. Let me open the whiteboard so I can discuss it with you how we do that. Okay. So the information I had was that this is maybe the white blood cell, and this white blood cell is coming up with 5 division of eyepiece graticule. So 1, 2, 3, 4, and 5. The problem over here is that I don't know the size or the division size of the eyepiece graticule. So the first thing which I need to know is I need to know the distance between the 2 division. So to know the distance between the 2 division, distance between the division of eyepiece graticule, between the division of eyepiece graticule, the first thing I have to do is remember, I need to do calibration. And I will do calibration with what? I will do calibration with the stage micrometer. Yeah. As many of you struggle with this calibration process and the exam, in the exam, the question comes up. So I'll go in little detail how this calibration is done. Okay. So for example, if I have this 5 division, 1, 2, 3, 4, 5. So I am provided with, this is your microscope, and inside the microscope, I have the eyepiece graticule with these division. The next thing I'm going to do is I'm going to use a stage micrometer. On a stage micrometer, a small line is present, okay? And this small line, sometimes they tell you that what is the size of the small line. Sometimes you need to know it by yourself. Normally, this line is 1 mm and it is divided into 100 divisions. So one division of a stage micrometer will be equal to 0.01 mm. Got it? So how we do this calibration? We do it in a way that firstly, what I'm going to do is, if this is my eyepiece graticule, I'm going to arrange the stage micrometer in a way that my first division of a stage micrometer completely coincides with the first division or with any division of the eyepiece graticule. So the two division, eyepiece graticule and stage micrometer divisions should completely coincide with each other. And then I will look for any other division which is completely coinciding with each other. Okay. So what I'm finding over here is the 4 division of eyepiece is equal to 1, 2, 3, 4, 5 division of a stage micrometer. So what I'm going to do is I'm going to say 4 division of eyepiece is equal to 5 division of stage micrometer. Because I know the size of the smallest division of this stage micrometer and it was 0.01 mm, so I can say that 4 division of eyepiece is equal to 5 multiplied by 0.01 mm, which will become 0.05 mm. And if I need to convert it into micrometer, I need to multiply it with 1000. So it will become, to get it into micrometer, I have to times it with 1000, okay? Times it with 1000. So if I look in the micrometer, my answer will become 50 micrometer, okay? So I got, if this was the size of, for example, if this was the size of my, any of my organelle, I expect 4 division, I can find the size of my white blood cell, or I can find in the same way, I can also find the size of 1 division of eyepiece, size of 1 division of eyepiece, and for that I have to divide 50 with 4. Okay, so it will be 1 and 2, 12.5 micrometer. Okay, so I just wanted to give you a brief know-how how we do the calibration. I'm going back to the original question now, okay? So I hope you have understood it. So when I go over here, so the first thing which I need to do over here is what additional information I need over here. My answer is going to be calibration of the eyepiece graticule using a stage micrometer. I'm going further ahead with the next question. Would the statement explain why it is necessary to use an electron microscope to see the crystal of mitochondria? Again, I'm given with the 4 choices: the magnification of the electron microscope, is greater than that of the light microscope. Always remember, when we are looking for a membrane, the size of the membrane is 7 nanometer. The resolution of light microscope is 200 nanometer. So we are going to focus more over here on the resolution, not on the magnification. So as we have to focus on the resolution, this choice will become wrong. The membrane of the crystal are separated by the distance greater than 200 nanometer. If it is greater than 200 nanometer, I should be able to see it with the light microscope. Okay. The maximum resolution of microscope using visible light is too low. Because the resolution of light microscope is less than 200 nanometer, we cannot observe anything whose size is less than 200 nanometer or the distance is less than 200 nanometer. So the wavelength of the electron beam is longer than the wavelength of electron beam is shorter than the wavelength of the visible light. Okay. So the answer over here is C. I'm going for question number 3 now. Some stains can be used to identify cell structure in living cell. A dilute solution of one stain causes the whole cell to appear blue. The blue color rapidly disappears from most cell structure. Those cell structure that releases energy stays blue. So I'm finding over here the keyword. There is a cell structure which is releasing energy and which is staying blue. I have been given with 4 choices over here: endoplasmic reticulum, Golgi body, lysosome, and mitochondria. What examiner is looking for over here is that can you identify that which organelle is somehow related to the energy? And we know that endoplasmic reticulum has nothing to do with energy. Golgi body does modification. Lysosome carries hydrolytic enzyme. So the only organelle which is related to the energy is mitochondria. And mitochondria were defined as a site of aerobic respiration. Why I'm writing site of aerobic respiration and what I'm trying to give you the key words also over here. Okay, most of the time the examiner do not accept energy and all that, so always remember mitochondria is a site of aerobic respiration. It releases ATP, and yes, ATP is energy. Okay, so mitochondria is going to stays blue. I'm going to the next question now. The next question is asking you, when mucus is secreted from a goblet cell, these events takes place. Addition of carbohydrate to protein, fusion of vesicle with the cell surface membrane, extracellular release of the glycoprotein, and separation of the vesicle from the Golgi body. I'm going one by one with the process. This is your nucleus. Messenger RNA comes out of the nucleus as we are talking about the mucus. Mucus is basically a glycoprotein. And I find endoplasmic reticulum. Endoplasmic reticulum synthesizes protein, and the small vesicles pinches off from the endoplasmic reticulum. If I look at the choices over here, they have not given any choice of endoplasmic reticulum. It means they are talking about the events which is happening afterwards. So as the vesicles pinching off from the endoplasmic reticulum fuses, they make Golgi apparatus. So this is the Golgi apparatus, closed structure. Okay. Now, what happens in the Golgi apparatus? In the Golgi apparatus, proteins get modified, and one way of the modification of the protein is that in the Golgi apparatus, a carbohydrate chain get attached with the protein, as we are talking about the mucus, which is a glycoprotein. So over here, the first process, which is showing over here, is going to take place in the Golgi apparatus. as the glycoprotein is going to be made, small vesicles are going to pinch out of the Golgi apparatus. And this is my fourth. Okay, and these small vesicles carries Golgi. These small vesicles carries glycoprotein. In the next stage, these small Golgi vesicles are going to fuse with the membrane, which is over here. Fusion of the vesicle with the membrane, which is the process 2. And then finally, separation. Yes, fusion of vesicle with the cell surface membrane, which is 2, and finally the extracellular release of glycoprotein, which is 3. So if you look at the sequence, I'm finding 1, and then I'm finding 4, and then I'm finding 2, and finally 3. So my answer is going to be A. I'm going further ahead. The diagram shows 4 biological features: DNA, RNA, 70S ribosome, and capsid. Which biological features are present in typical prokaryote? So over here, precisely, examiner wants to know that, can you differentiate between a virus and a prokaryote, which is a bacteria? When I look at the virus has a protein coat and a genetic material, and this protein coat is called capsid. However, when I look at the bacteria, the outer cell wall is made up of peptidoglycan. So for sure, capsid is not present in bacteria. So as capsid is not present in bacteria and it is 2, so this is carrying 2 cancel, this is carrying 2 cancel, this is carrying 2 cancelled, and we are left with the choice of C. So yes, the bacteria does carry 70S ribosome. It carries DNA as a genetic material, and it carries messenger RNA and tRNA also. So C will be our correct answer. I'm going for the question number 6 now. Question number 6, which row shows features that occur in dicot plant cell and also in typical bacterial cell? It means that the examiner is looking that, can you differentiate between a eukaryote cell and a prokaryote cell? Because plant cell is basically a eukaryote cell. Okay. So when and the examiner is looking for one more thing, not only for eukaryote cell. If it is a eukaryote cell, can you differentiate between the animal eukaryote cell and a plant eukaryote cell? So when I look at yes, both the bacteria and the dicot plant carry 70S ribosome, and in plant cells, 70S ribosome is present in their chloroplast. In bacteria, there is no 80S ribosome. Okay, so it is not present in both. Centrioles definitely not present in both, and circular DNA is present in both, in prokaryote as well as in plant. Again, in plant, this circular DNA is present in the chloroplast. So, if I look at the choice B, yes, 70S ribosome is present in both. 80S ribosome is not present in both, which is correct. Centriole is not present, and the circular DNA is present in both. Okay, so the answer is going to be, so the are not found in at least one of these. Okay, centrioles are also not present in the plant cell. There are some plant which are the advanced plant where you may find the centrioles. Otherwise, centrioles are not present in the plant cell. So no. Okay, so the answer is going to be B. I'm going for question number 7 now. Which flow chart outlining the test for non-reducing sugar is correct? Okay. Okay, Now, for that also, we need to work on that. Can we differentiate between reducing sugar and non-reducing sugar? We define reducing sugar as all monosaccharides and disaccharides are reducing sugar except sucrose. So when I look at the sucrose, which is a non-reducing sugar, sucrose is made up of glucose plus fructose. Okay. So, for example, if I apply a Benedict test and Benedict's test stays blue, it does not changes the color. So it means that the sample does not carry the reducing sugar. So I want to know that does the sample carries non-reducing sugar? So what I'm going to do first, I will take an unknown sample and I will boil it with HCl. So I'll boil it with HCl. Why I am boiling it with HCl? Because if the sample carries sucrose and I boil it with HCl, sucrose is going to break down into or hydrolyze into glucose and fructose. In the next stage, I'm going to neutralize this HCl and I'll apply Benedict test. If it was sucrose and it got broken down, it got hydrolyzed, it means that the sample carries glucose and fructose. In that case, if I apply Benedict test, my result is going to come positive. Okay, so my answer is going to come as C. The confusion may lie with C and D because both are using HCl, both are neutralizing. But this is here they are using Benedict and here biuret. Remember, biuret test is for protein. It is not for reducing sugar. Okay, so my answer is going to be C. Now I'm coming with question number 8, which is asking me which diagram shows the release of water molecule during the formation of glycosidic bond. So the first thing which I need to know over here is that glycosidic bond is made between sugar. Okay, so at first I need to identify the molecule they are showing. Are they sugar? So yes, I can see they are sugar and I can see all of them are hexose sugar. Okay, I'm giving with the choice over here. Which one of them is so during the formation of which diagram shows a release of water molecule. Okay. Choose the release of water molecule during the formation of glycosidic bond. Okay, so I can see as the condensation is happening between the two, water is getting released over here. The condensation is happening over here too, so the water is releasing over here. And over here also, the condensation is taking place. So in all three of them are correct over here. Water is getting released, all of them showing me the process of condensation between a carbohydrate molecules. Question number 9. Which molecule have a structural formula that contains C double bond O? Okay, so when I look at the C double bond O, we need to know that if I look at the amino acid, if I make the simple diagram of amino acid, it is C double bond O, OH, and over here there's an H, and over here there's a variable group or R group, and over here I have NH2. So I can see C double bond O over here, okay? Similarly, as it is fatty acid, so yes, fatty acid definitely carries COOH with variable chain, okay? Glycerol molecule does not carry any double bond. It is a 3-carbon structure with OH on all the sides over here with H, 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 and H, okay? So I can see that I found C double bond O in amino acid. I found C double bond O in fatty acid. And as protein is also made up of amino acid, so I will find it in protein. So the choice will be 1, 2, 4, and the answer is going to be B. Okay. I'm going further ahead with question number 10. Jaden, any question till here? Are you able to understand it well?
Yeah.
Okay. I'm going for question number 10 now. Which statement about triglyceride is correct? Triglyceride. Triglyceride means a glycerol molecule with 3 fatty acid chains. So a glycerol molecule with 3 fatty acid chain, which can be same, which can be different, which can be saturated, which can be unsaturated. So in any triglyceride, all the fatty acid are saturated or all fatty acid are unsaturated. That's wrong, because this fatty acid can be any fatty acid. Each chain is very, very independent. Okay, so it can be saturated, it can be unsaturated. There is a possibility that all the 3 chains are saturated or all the 3 chains are unsaturated. So over here we have all the 4 possibilities with us. So we cannot stick up with one of the possibilities. Question number, the choice B is saying the 2 triglycerides are joined together by an ester bond. So no, ester bond we find between glycerol and the fatty acid. Over here I find the ester bond. See, triglyceride are polar hydrophobic. Now here, basically examiner wants you to differentiate triglyceride with phospholipid. Yes. Phospholipid is a molecule which has a polar head and a non-polar side, but triglyceride, there is no uneven distribution of charges. It cannot be divided into polar and hydrophobic. So we are left with the choice D, and D is basically a glycerol, and glycerol is joined by fatty acid by covalent bond. Okay, this is basically a covalent bond. Now comes question number 11. Many flower produces a sweet solution called nectar. Bees provided with nectar use enzyme Q to change the nectar into honey. So what I'm providing with the information, I have an enzyme Q, which is converting nectar into honey. The next set of information I'm having is, after testing a sample of nectar for presence of reducing sugar, using a standard laboratory reagent, the sample was blue. That tells me that the nectar carries non-reducing sugar because Benedict taste test came blue. However, when I check honey, honey test positive for reducing sugar because it is orange, so honey carries reducing sugar. Okay, so it means that the sucrose somehow got broken down, and to break down something, we always use hydrolysis. So these choices completely got wrong. I'm left with the choices hydrolysis, C and D. Now, as we have already mentioned over here that nectar carries non-reducing sugar, so yes, sucrose it is carrying, and the product is carrying fructose, which is a reducing sugar. So we have identified already over here that nectar was carrying sucrose because sucrose is the only non-reducing sugar. Okay. Secondly, D is also wrong because maltose you get by the breakdown of the starch. Okay. So, and maltose is a reducing sugar. The next question they're asking you is question number 12. Little tricky, but a very easy question, okay? If you understand the structure, it becomes very easy for you. So the question they're asking you is, the diagram shows the structure of the part of the peptidoglycan molecule. So I can see sugar and I can see an amino acid chain, a polypeptide chain. What they're asking you is which type of 1,4 linkage? and how many peptide bonds are present. So first we need to look for what type of glycosidic linkage I can see over here. So the glycosidic linkage I can see is, yes, I can see the linkage is being made between 1 and 4. And if I identify the sugar, I can find it as beta sugar. Why? Because hydrogen I can see down. Okay, in alpha, hydrogen is up. So this linkage is going to be, beta 1,4 glycosidic bond. The next thing which examiner wants us to see is how many peptide bond. To understand how many peptide bond, we need to know how the peptide bond is formed. And it is formed between which two molecules. So if I make an amino acid, peptide bond is formed between NH2 group of 1 amino acid and COOH of another amino acid. Condensation takes place and this water molecule has been removed. So as this water molecule is going to be removed, creating a peptide bond, it means that your nitrogen is going to left with one hydrogen. So I need to look for NH. Wherever I'm finding NH, that is going to be the point where condensation is taking place. So I'm finding NH over here. I'm finding NH over here. I'm finding NH over here and I'm finding NH over here. So let's see the beta 1,4 glycosidic bond and 4 a peptide bond. So my answer is going to be D. Beta 1,4 linkage and 4 peptide bonds. I'm going for question number 13 now. And the question number 13 is a little more information over here. Let's see it. I'm provided with a graph, time versus percentage absorbance. So a scientist investigated the progress of two enzymes catalyzed reaction in separate test tube, X and Y. Both the reactions result in color change that can be detected using colorimetry. 0.5 cm cube of sample were taken from each test tube at the start of the investigation and at a regular interval for the next 5 minutes. Copper ions were added to each sample as soon as the samples was collected to inactivate the enzyme and stop the reactions from processing further. So copper ion is acting as an inhibitor. So we are using two enzyme X and Y, and we are what we are recording is the percentage absorbance. So now let's see what choices I'm given with. Which statement is consistent with the result shown in the graph? Okay. The first choice is telling me the substrate in test tube X has a higher absorbance than the product. So see over here, I have a if I see over here, I have a substrate and X, and here I have a product. So this simply becomes wrong because I find higher absorbance at the product end rather than the substrate end. So this is wrong. B, the product in test tube Y has a lower absorbance than the substrate. Yes. This has, Y is showing me higher absorbance over here. And over here, the product has less. So yes, this seems to be correct. C, the rate of reaction in test tube X increase. I have no information about the rate of reaction. And over here also, this graph is not telling me any information about the rate of reaction. So the direct, when I look at the graph and they ask you to deduce the answer by the information given, look for the direct information which you can derive from the data or from the graph. You do not have to make any assumption. Over here, yes, I can make the assumption that as uh, the absorbance is increasing or decreasing, some rate of reaction must be happening, okay? But I cannot deduce it directly. Secondly, it is not consistent because after that, I do not see any change. After this also, I'm not finding any change in the rate of reaction. However, this time, they are talking about the overall time between 0 to 5 minutes. So after this, there's no change in the rate of reaction. So I cannot go for the choice C and D. We will go for the choice B. The next question is question number 14. I have given with some choices. A student investigated the hydrolysis of lipid in high fat milk using the sample lipase. So I have a fat and I have an enzyme lipase, okay? And how I'm finding the rate. So I have added high fat milk, temperature I'm keeping constant, and I'm measuring the pH to know the rate of reaction or something. Now, why am I measuring the pH? Because when the fat got broken down, it got broken down into fatty acid and glycerol. And more fatty acid means the pH will keep on decreasing as the reaction proceeds. Now they have given me 3 statements and I need to describe the expected result. Which statement correctly describe the expected result? So the first statement is telling me the product forms more slowly as the time proceeds because the concentration of substrate is decreasing. I have no information of this. Which statement, although I have no information about this, but I know that this happens because as the substrate concentrate, enzyme activity is affected by 4 factors: substrate concentration, enzyme concentration, temperature, and pH. So yes, I can describe, because over here they're asking you describe. Yes, so it is correct. The product which is going to be formed slowly. I should get a reaction like this because the concentration of the substrate is going to be decreased as the reaction proceed. Okay, then comes question 2. The pH of the reaction mixture increase. Reaction mixture pH cannot increase because the fatty acid is going to decrease the pH. So this is wrong. The third one is the increase in the concentration of product eventually causes the lipase molecule to denature. Yes, because this is going to cause the change in the pH. And as the pH will keep on decreasing, a time will come when the enzyme will start to get denatured. So my answer over here is going to be C. I'm going for the question number 15. What is the correct range of measurement in the width of the cells? This is just the question of not understanding. You just need to know it, okay? So we know that if I look at the thickness of the membrane, on average, we take is 7 nanometer. As a range, we take it between 5 to 10 nanometer. The next question they're asking you is where in the cell surface membrane are the carbohydrate chain of glycoprotein and glycolipids mainly located? So I'm drawing a phospholipid bilayer. This is how your membrane looks like, and here is the glycoprotein or glycolipid. Okay. When I look at these protein, I can see that we always make the chain on the outer side. Why the chain has to be on the outer side? Because of these glycoproteins are acting as a receptor for signaling molecule. So they have to receive anything from on their surface. So receptors. Similarly, they may be acting as an antigen for cell-to-cell recognition. So again, they are required to be on the surface, not on the inner side or towards the cytoplasm. So the answer is going to be D. You will find glycoprotein as well as glycolipid on the outer surface. The reason is because of the glycoprotein and glycolipid. These proteins are acting as a receptor to accept the signaling molecule. They are acting as an antigen to do cell-to-cell recognition. And all these things are going to be happen on the outer side of the membrane, not on the inner side of the membrane. Then comes question number 17. Question number 17 is saying sodium ion can enter cells across the cell surface membrane. Now over here, the key word is ion. Okay, so it means that for sure, for ion, I will be requiring some kind of carrier protein. Okay. Now, the next thing which is going to decide that is it going to happen by active transport or by facilitated diffusion, that is going to be decided by the concentration difference. If it is moving from high concentration to low, it will be facilitated diffusion. If it is happening other way around against the concentration gradient, it will be active. So only active transport is wrong because it depends on the concentration gradient. It can happen through active or facilitated. Simple diffusion is not possible at all. Simple diffusion is not possible at all because it is an ion. An ion cannot pass through the membrane by itself because hydrophobic interior won't let it pass through. It is a charged molecule. We are going now ahead with the question number 18, which is relatively easier question. They're asking you, they have given you a beaker. In the beaker, in one of the beaker, I can see 5% sodium chloride solution. In the other beaker, I find a 10% chloride solution. Just give me a moment. Okay. So what they're asking you is they're asking you which row correctly describe and explains what will happen if in the half of the beaker labeled S. So if I see over here, S is a beaker with more concentration of sodium chloride. So I can deduce that it has a low water potential. And this one has a high water potential. Okay, this is a sign of water potential. Okay, so as it has a low water potential, so it is very obvious that the water is going to move in this direction. Okay, now let's see what choices I'm provided with. I'm provided with 4 choices. Volume of the solution in the S increases. Yes, it will increase because water is moving. Because net movement of water from a high water potential to low water potential. So my answer is going to be A. Okay. Over here, this is fine. Volume of solution increases, but they are saying net water movement from low water potential to high. That is not possible. Volume of solution in S decreases. That is going to be wrong. And the volume of solution in S decreases is wrong. So I am left with the choice of only A. The next question is question number 19. Agate cubes can be used to demonstrate an effect of diffusion on changing the surface area to volume ratio. So one thing you need to remember that whenever they give you this question, they are looking for Fick's law. And Fick's law tells me 3 things. That the rate of diffusion depends on, number 1, concentration gradient, and it is directly proportional. Number 2, just wait. Concentration gradient. Number 2, surface area to volume ratio, again directly proportional. And number 3 is distance of diffusion. It is inversely proportional. As the distance increases, rate of diffusion decreases. Okay, so we can conclude that if some structure is smaller, the rate of diffusion is going to be faster. Over here, they have given you a cube and they told us that the size of the cube is 2 centimeter cube. And we are dipping it in some solution and that solution is diffusing in, which is giving it a color. Okay. And they're asking you which diagram shows the result for 1 cm cube and the 3 cm cube. Now, let's first go for elimination process. If I go for the elimination process, that is not possible, that the size is small and the diffusion has not taken place at all. So A will become completely wrong because I cannot see any diffusion over here. B will also become wrong because they have given you, just wait, okay. B will also be wrong because I cannot see any diffusion over here. Now I'm left with the choice C and D. Okay. When I look for the choice C and D, I see that in C, the diffusion has taken place. Yes. And over here also diffusion. Okay. So, and over here I can see the diffusion, and yes, diffusion is also taking place over here. So my answer is going to be C. Okay, because these two are wrong. Okay. Over here, if I compare it with the size, this is showing me too small over here. So if I compare it with 2 cm cube, what I'm getting with 3 cm cube, my answer is going to be C, which is the closest one. Okay. Okay. Then comes question number 20, and the 20 is saying during sperm formation in mammals, part of the structure of each chromosome is replaced with protein called protamine. The replacement allows the DNA to be packaged much more densely than what otherwise be possible. Which part of the chromosome is replaced by protamine? So something we are replacing, which is already causing the packing. So remember, if I open one of this cell and I get into your nucleus, I have 1.8 meter long DNA. Okay. To occupy such a long DNA, we wrap it around a protein molecule, which is called histone. Okay, so histone is used for packaging. So as your question is already indicating that you are replacing something which is being used for packaging. So the only molecule which we use for packaging is going to be histone. So my answer is going to be C. Got it? So I'm going further ahead. Jaden, any question here?
No.
Is it helpful?
Yeah.
Okay, great. I'm going ahead now. So I'm going for 21 now. Here. The diagram shows the mitotic cell cycle, during which phase do chromosome condense and become visible. So what examiner is looking for? That are you able to differentiate the cell cycle into interphase, mitosis, and they have already given you cytokinases, okay, which is the third phase. So if I look at over here, I can see that these three phases, which is G1, S, and G2, it comes under interphase, and D is mitosis. So when I look for mitosis, in the first phase, prophase, the condensation process takes place. So my answer is going to be D. Okay. I'm going to the next question now, which is question number 22. 22 is asking which statement correctly describes the feature of the stem cells that is essential for their role in cell replacement and tissue repair. So I'm over here, I'm providing with 3 keywords. I'm finding a stem cell, and I want to find the feature of the stem cell which helps it in cell replacement and tissue repair. Okay, it is tissue repair. So the first one is saying me, after mitosis stem cell, the daughter cells can either remain as a stem cell or follow a development pathway that leads to the formation of a specialized cell. So yes, this is your stem cell. It divides. For example, I say that this stem cell is present in your bone marrow. And in bone marrow, different blood cells. So it got differentiated into red blood cell. It has multiplied. It has made cells, which has developed into red blood cell, different kind of white blood cell, platelets. Okay. And then there are some stem cells remain over there because your RBCs have a life of only maximum of 120 days. You need to make it continuously. So the stem cells has to be present over there, okay? And then this stem cell has been also been used in a specializing. So yes, one seems correct. When I go for the second one, it is saying stem cells are different to all body cells because they retain all the genetic information in their DNA throughout the life of an organism. Remember, as we have developed from zygote, and the zygote has multiplied by mitosis. So whichever cell I pick up from your body, either it is a lung cell, or it is a heart cell, or it is a stem cell, the genetic material is going to be identical. Okay, there will be no change in the genetic material. And each and every cell of you, except few exception like red blood cell, have this genetic material. So 2 will become wrong. Then I'm going for 3. A small proportion of a stem cell is retained in the body of adult throughout. I have given you the example of stem cells in your bone marrow. They stayed in your body all your life. And the 4th is the stem cells have more telomeres. No, telomeres are present at the end of the chromosome, and the number of chromosomes, there is no change in the number of chromosomes in any of your cell of your body. Okay, so 4 will also be wrong. I will left with the choice of 1 and 3. Okay, I'm going for question number 23 now. And 23 is asking me which statement are correct for all nucleotides. So if I say nucleotide, so your DNA is a nucleotide, your RNA is a nucleotide, and your ATP is also a nucleotide. Okay, so let's see what is common in all the nucleotides. The nitrogen containing base is always attached to the carbon atom of the pentose sugar. So if I make, for example, I'm making a DNA molecule. So this is your ribose sugar or deoxyribose sugar, or if I talk about ATP, again, this is ribose sugar. I can see it is attached to carbon number 1. So this is correct. Yes. Okay. The second is the phosphate group is always attached to the carbon atom 5 of the pentose sugar. Not necessarily. Not necessarily. This phosphate can run in 2 directions. We have already done. It can attach to carbon number 3, or it may attach to carbon number 5. So as they say always, this becomes wrong. A third is saying me a condensation reaction occurred to join nitrogen containing base to the pentose. Whenever we want to join something, we have no other option than condensation. So this is also correct. 4th, nucleotides are linked together by condensation reaction between nucleotides. If I talk about the DNA, and I talk about the double helix DNA being attached with each other, they're attached with hydrogen bonds. Nucleotides. So they are between the phosphate group. And if I talk about this one, this nucleotide, one nucleotide with another nucleotide, that is sugar phosphate backbone. So condensation is happening between sugar and phosphate. So this is also wrong. So I will left, just let me see this again. Nucleotides are linked together between phosphate group. So let me see again. So 1, 3, 4. I'm revisiting it. Sorry, I'm revisiting it. When I look at this nucleotide are linked together between phosphate group. So it comes like this, phosphate, and over here, phosphate. So yes, we can say that the condensation is happening between sugar phosphate, but the nucleotides are linked together by phosphate group. Yes. Okay. So my answer is going to be 23 is going to be C. Yes. 1, 3, and 4. Okay. Now I'm going to question number 24, which is again a little tricky question. 23. To the pentose sugar. A condensation reaction occurred to join the nitrogen-containing base to the pentose sugar. This is correct. Yeah, this is correct. So 1 and 3, I need to revisit it here. Let me check it again. So this is 3 is correct. And the phosphate group is always attached. Oh no, that is wrong. Oh, come. Okay, let me revisit it. 23. Okay. Carbon atom of the 5 of the pentose sugar. So, okay, sorry. I'm going to revisit it. Let me erase it. I need to see it again. Just let me, I think so I have made some mistake here. Let me revisit it. Okay. Here we are. Which statement are correct for all nucleotide? I'm going ahead with this. Let me take true. Okay. Which statement is correct for all nucleotide? The nitrogen containing base is always attached to the carbon atom. This one was correct because we have made this ribose or deoxyribose sugar. So this is correct. I'm going for the next one. The phosphate group is always attached to the carbon atom 5 of the pentose sugar. The phosphate group is always attached to the carbon number 5. Yes. In one way, I can say that carbon number, okay, let me see it. If I see it as an ATP, this is carbon number 5, okay? So fine. If I look at the DNA or RNA, so it comes like this. Here, here, and here, and here. So yes, over here also, if I look at the nucleotide, the phosphate group is always, if I look at the nucleotides and I see the single nucleotide, not as a whole, if I see a single nucleotide, I was taking it as a complete chain and there I was making mistake. I need to consider it as a single nucleotide. So as a single nucleotide, 2 is also correct. Okay, and then comes the condensation reaction. So condensation reaction is happening between a pentose sugar and a nitrogen containing base, which is 3. So the answer is A. Where I have made the mistake, my mistake was that I was taking nucleotide as a DNA molecule. They are talking about single nucleotide. Okay, so I need to consider single nucleotide. My answer is going to be A. I'm going with the question 24. 24 is asking me, how many phosphodiester bonds are present in circular DNA molecule of 2700 base pair? Phosphodiester bond containing 2700 base pair. So if I look at the phosphodiester bond, so even if I look at because it's a base pair and circular DNA is also a double helix structure. So it is 2700. I have 2 of them. So it makes 5400. And for each chain, there will be 2700 phosphodiester bond, and I have 2 chains. So my answer is going to be 5500. Any question here? Jay,
No.
any question here?
No.
Okay. Okay, dear. So over here, I'm stopping it, and the rest of the questions we are going to do in our next class. Is it okay with you, Jaden?
Okay.
Okay, dear. See you in the next class. Bye.
Bye.